Today, we will see transcription in eukaryotic. Eukaryotes are multicellular organisms like animal and the plant, fungi and algae. How the process of transcription is happen in this kind of organism. So, if you are already member of this Nugu Academy, like and share this video. If you are not a member, subscribe the video. Let us continue. What do you mean by transcription in eukaryotes? How this process is initiated, like initiation, elongation, and termination. Before we see the details, what do you mean by transcription? Transcription is the process by which information contained in DNA is converted into that of RNA. There are nitrogenous base sequences in DNA. That nitrogenous base sequence could be converted into RNA. That process is what you call transcription. But in eukaryotic cell type, the process is very, very complex. There is a nucleus. In the nucleus, there is a DNA. The process is very, very complex. The cell is larger. It's not simpler like that of prokaryotic. In prokaryotic, once the process of DNA replication happens, transcription proceeds. The process is coupled. Then immediately the process of protein synthesis happens. But in eukaryotic, it is very, very complex. It is very, very complex. How it is initiated, how synthesis is carried out, and finally, how it is terminated. All these processes carried out in nucleus. During initiation, there are RNA polymerase 1 and RNA polymerase 3. It transcribes gene encoding RNA, particularly ribosomal RNA and transfer RNA. RNA polymerase 2, it transcribes gene encoding protein. It converts the sequence written in DNA language into RNA, then it codes for protein. So we'll see this RNA polymerase 2. How that sequence is converted, how it codes for the protein. For this matter, as I told you, 3 stages are very, very important. Initiation, elongation, and termination. What happened at the initiation stage? There is a region we call a promoter. This promoter region is from -35 to -10 region. It contains a kind of sequence, recognition sequence and binding sequence. For example, at the -30, at the -9 region, Tata box sequence, it is binding region. It is a binding region like prokaryotic. The question is, how does RNA polymerase find this particular sequence, or we call it promoter? This is the question. For this matter, just like prokaryotic, transcription factors are very, very important. There are transcription factors that aid RNA polymerase. In this case, transcription factor D, transcription factor A, transcription 2 factor B, transcription factor D, transcription factor E, F, and transcription factor, they form what we call pre-initiation complex. They aid RNA polymerase 2 to recognize that region, and they form what we call pre-initiation complex. With the help of these transcription factors, RNA polymerase 2 able to bind at the -35 region of recognition. Then it forms a complex at -10 region. We call it Tata box. For example, transcription factor D, it contains Tata binding subunit or a domain. We call it Tata binding protein. This Tata binding protein or transcription factor D, it helps recognition of a promoter, or it serves to recruit target transcription factors to bound to upstream promoter elements. Transcription factor A, it's not essential. It's not essential for pre-initiation complex function, but it serves to stabilize binding of transcription factor D to the DNA. Transcription factor D, it has a role in binding to DNA. Transcription factor E, it recruits, it builds or brings transcription factor H to pre-initiation complex. Then it forms a kind of a complex. Transcription factor F, it recruits RNA polymerase 2 to the position. That position is pre-initiation complex. So it forms a kind of pre-initiation complex. It means that initiation is begun. Once initiation is started, the process of transcription starts at +1. At a +1. Then elongation process go ahead. In elongation process, the polymerase is released from other transcription factors and pre-initiation or pre-messenger RNA synthesized in a direction of 5 to 3 direction. Then polynucleotide synthesis to occur, transcription machinery need to move histone out. Then it carried out what we call elongation. Mind you, in case of eukaryotic, the process is very, very complex because, in nucleus, there is a DNA and a histone. DNA and histone they are wrapped around each other. They are twisted. So, if that histone is not removed, transcription never begins. For that matter, transcription machinery needs to move histone out of that point. That machinery is what we call FACT or facilitates chromatin transcription. DNA plus chromatin is called histone. Now, histone is removed with the help of the factor we call a FACT, facilitate chromatin transcription. It pulls histone away from DNA template as RNA polymerase moves along it. In such a way, synthesis of the strand carried out. Then after synthesis, it stops the transcription. That is what you call a termination. How it is terminated? Elongation of RNA chain proceeds as long as polymerase encounters stop signal. There is stop signals at the end, especially at the 3' end. At the 3' end, it encounters that signal, especially AA, UU, AA, UAAA, that is polyadylation signal. When it encounters this signal, it stops transcription. It means that this signal tells RNA polymerase to stop what we call a transcription. But the actual termination of transcription by RNA polymerase 2 occurs beyond the transcription or polyadylation site. There are three factors for this matter: cleavage factors, CPSF factors, or CSTF factors. CPSF, it is cleavage and polyadylation specificity factors. CSTF, cleavage stimulatory factors, again cleavage factors. It is these factors that aid the termination process. Look at here. This is strand from 5 to 3 end. You know there is a C terminal. In nucleolase, this C cleavage and polyadylation specific factors, it moves from nucleolase to this region. The region we call polyadylation. AA, U, AAA. It binds at this region. It binds at that region. Cleavage stimulatory factors moves to CA region. CA region is found at the right away of polyadylation. It binds at this region. Cleavage factors cleaved at the sequence of CA. It means that it cut and release messenger RNA. Finally, during RNA processing, sequence downstream of polyadylation signal is removed. By such process, messenger RNA synthesis terminated. So it is these factors, this RNA polymerase, here CPSF, CSTF, cleavage factors. It is these factors that aid the termination of the transcription. What are the mechanisms? The mechanism is actually, it is unknown. Thank you for watching this video. Subscribe, share, and comment. Thank you very much.